Hello everyone, welcome to the new session of chapter 15, plant growth and development exercise. So today we are going to solve the NCERT questions from the chapter plant growth and development, right? So let us start the session. Okay, so in this session we will start with the NCERT question 1 here. So the question 1 is define the growth and differentiation, development, de-differentiation, redifferentiation, determinate growth and meristem and growth rate. So all the definition we have to give in this particular question. So let us see. First, the definition of growth is here. It is an irreversible and permanent process. So growth is a irreversible process. Okay, so growth can never be reversible. So it is an irreversible and it is a permanent process accomplished by an increase in the size of an organ or organ parts or even of an individual cell. Right. So how the growth is occurring? The growth is occurring by increase the size of an organ or organ part. Any organ can increase its size, or any organ part, one part of the organ can also increase in size, or even an individual cell. The size of the cell get enlarged, though then also you can say that it is a growth, and this growth can never be reversible. It is always irreversible process, right? Next definition is the differentiation. What is the meaning of differentiation? What is the definition of the differentiation? So it is a process in which the cells derived from the apical meristem. So there are apical meristem, okay, at the two apex, one at the shoot apex and one at the root apex. So root and shoot apex, and the cambium that undergoes structural changes in the cell wall and the protoplasm and becoming mature to perform the specific function. So this is called as the differentiation, right? So what is occurring in the process of this apical meristem? Apical meristem undergoes divide and redivide and form the new cells. So the new cells are derived, right? And also the cambium is doing the same thing, that the cambium layer that is present in the vascular bundle, it divide and redivide and forms the new cells, right? So it undergoes structural change. The new cells which are formed, it undergoes the structural change, right? The cambium, the apical meristem, these two cells undergo the structural change. How it is going, the change? Cell wall and protoplasm. It changes its cell wall and protoplasm and becoming mature to perform specific functions. Now, the development. It refers to the various change occurring in an organism during the life cycle from the germination of the seeds to the senescence, right? So development is here. So it is the definition of the development, that organism, whatever the changes that you see in the life cycle of an organism. So there is a seed. When the seed germinate, it gives rise to a new plantlet. The plantlet get established in the soil, and later it forms a new big plant, right? And later what happened, when the unfavorable condition is there, some of the leaves get under the senescence process. Okay, it gets yellow and they later shed off even. So this is all the stages of the development. De-differentiation. It is the process in which the permanent plant cell regain the power to divide under a certain condition. Only some of the cells, they are not having the power of undergo the division condition every time. At a particular stage, right, they regain the power to divide under the certain conditions only. So that is the process of de-differentiation. Next definition, redifferentiation. What is the meaning, or what is the definition of the redifferentiation? So it is the process in which de-differentiated cells become mature again and lose their capacity to divide. Okay, so it is the process in which de-differentiated cells become mature. They again become mature. Earlier they were dividing, but now they become mature and lose the capacity to divide. Now they are not dividing. They got differentiated and they turn to the mature cell. So this process is called is redifferentiation. They are returning back to their original form. Only at certain condition they are under the division phase, but later when the condition is withdrawn, they again changes to the mature cell. So this is called as a redifferentiation process. Determinate growth. It refers to the limited growth. Okay, it refers to the limited growth. For example, animals and plants leaves stop growing after having reached maturity. Animals and plant leaves, they do not grow, show any type of growth type of things, right? So they are, when they reach to the maturity, they stop growing. So they do not show any enlargement of the cell, or the size of the leaves get enlarged. At a particular stage, they show this, but after certain stage, they stop the increasing the shape or the size of the leaves. So this is the determinate growth. Next is the meristem. What is the meaning of meristem? So in plants, growth is restricted to the specialized region where active cells divisions takes place. Okay, some of the cells are only actively, okay, and they are involved in the process of division of the cells. New cells are formed. All the cells are not involved, only some of the cells are involved. So such a region is called as meristem. So the region of the plant where these type of cells are present, so that region is called as meristem. And there are three different types of meristem: apical meristem, lateral meristem, and intercalary meristem. So there are three different types of meristem. Okay, apical meristem, lateral meristem, and intercalary meristem, depending upon the position. So apical meristem is present at the two apex of the plant, that is the shoot apex and the root apex. Okay, lateral meristem is present in the vascular bundle, that is in the form of cambium, and at the time of secondary growth they form the whole ring, right? And intercalary meristem, they are present in the axils, at the nodal region from where the new branches arises, new axillary buds arises at the axils of the leaves. So that is the intercalary meristem. Okay, next definition is about the growth. What is growth rate? It can be defined as the increased growth in the plants per unit time. So you will take a unit time and you see whether how much the plant is undergoing the growth, right? So that measurement gives you the growth rate of that particular plant. Okay. Okay, so here comes the next question. Why is not any one parameter good enough to demonstrate the growth throughout the life of a flowering plant, right? Only one parameter they are talking about. Just by using only one parameter, you can demonstrate the growth of the plant throughout their life. Okay, is it possible? No. But why? Why it is not possible? In plants, growth is said to have take place when the amount of protoplasm increases. Okay, when you see a cell, the protoplasm of the cell increases, it increases the size, right? So measuring the growth of protoplasm involves many parameters such as weight of the fresh tissue. If the amount of the protoplasm increases, it increases the weight of the fresh tissue sample, the weight of the dry tissue sample, the differences in the length, area, volume, cell number measured during the growth period. Number of the cells also increases during the growth period. Volume of the cells increases. Area, size of the cell increases. The area it increases. Enlarges. The cell also enlarges. The length is increases. And you are taking the consideration of only the protoplasm increase, right? Only one parameter you are taking. So measuring the growth of plant using only one parameter does not provide the enough information, because so much things are just changing by increase of the protoplasm, the size, the area, the mass, the volume, right? The number of the cells, everything is changing. So why to consider just one parameter? It will not give you the exact growth of that particular plant. So measuring the growth of plant using only one parameter does not provide enough information, and hence it is insufficient for the demonstrating growth. Okay, it is insufficient for demonstrating the growth of the plant. Next question. Describe briefly arithmetic growth, geometric growth, sigmoid growth curve, and absolute and the relative growth rates. Okay, so these are the differences that you have to, these are the points that you have to describe briefly. So let us see the answer. Arithmetic growth. What does this arithmetic growth mean in the case of plant growth? So in arithmetic growth, one of the daughter cell continues to divide while the other differentiates into maturity. When the new cells are formed, so they are called as the daughter cells. Now some of the daughter cells get differentiates into the other type of cells. Okay, it continues to divide, right? Daughter cells continues to divide, whereas some of the cells differentiates to maturity. They become the mature cells and they do not divide, because the mature cells do not divide. So some of the cells, new cells that are formed after the division, okay, in arithmetic growth, they divide and redivide, but the other cells, they becomes mature. The elongation of the root at a constant rate is an example of the arithmetic growth. Okay, this is one of the example that is showing the arithmetic growth, that is the elongation of the roots. Okay, geometric growth. What is the meaning of geometric growth? Geometric growth is characterized by a slow growth in the initial stages and rapid growth during the later stages. Okay, so geometric growth, when you see the geometry of the cell, you see the growth is characterized by the slow growth. Initially, it shows a very slow growth, but later they show the rapid growth during the later stages. So the daughter cells derived from the mitosis retain the ability to divide, but slow down because of the limited nutrient supply. When the supply is less, okay, when the nutrient supply is less, they slow down their division. Okay, they do not divide, but when the nutrient supply is enough, they divide by the process of mitosis and increase their number. So this is showing the geometric growth. Okay, next is the sigmoid growth curve. The growth of a living organism in their natural environment is characterized by S-shaped curve called the sigmoid growth curve. So in the nature, you see such type of growth. Okay, sigmoid growth. Sigmoid means S-shaped growth. So this curve is divided into three phases: lag phase, log phase, and exponential phase of the rapid growth, and stationary phase. Okay, so the three phases there, that is the lag phase. This is the lag phase. This is the exponential phase, and this is the stationary phase. So initially in the unit time, the size weight of the organ is not increases very fast. Okay, it takes a time to show the growth. So that is called as the lag phase. That is called as the lag phase. But later you see the increase in the size weight of the organ. Right, at this particular stage of their life, they show very fast growth. So that is the exponential phase of their life. And after that, they again slow down their growth and they become is stationary. They do not it shows any increase in the size or the weight of the organ. So that is showing the stationary phase. So S phase we see in the nature of any organism. So exponential growth can be expressed as W1 is equal to W0 e to the power RT, where W1 is the final size, that is the final size of the organ, and W0 is the initial size, R is the growth rate, T is the time of the growth, E is the base of natural logarithm. So this formula is used to take out the growth, exponential growth. Absolute and relative growth rate. What is the meaning of absolute and relative growth rates? Absolute growth rate refers to the measurement and comparison of the total growth per unit time. Relative growth rate refers to the growth of a particular system per unit time and expressed on a common basis. Okay, so absolute growth rate refers to the measurement. You take the measurement, then you get the absolute, and comparison to the total growth per unit time, right? And relative growth rate refers to the growth of a particular system per unit time. List 5 main groups of the natural plant growth regulators. You have to name the 5 groups of the natural plant growth regulators. Write a note on discovery, physiological functions, and the agricultural, horticultural applications of any one of them. So here we are having the 5 different groups of the plant growth regulators. So the plant growth regulators are the chemical molecules secreted by the plants affecting their physiological attributes of the plant, physiological activity of the plants. And there are 5 main plant growth regulators. So we are having 5 main plant growth regulators. What are those? Auxin. Number 1 is auxin. Number 2 is gibberellic acid. Number 3, cytokinins. 4th, ethylene. And 5th is the abscisic acid. So we have to tell about the discovery, right? The first observes regarding the effect of the auxin were made by the Charles Darwin and Francis Darwin, where they saw the coleoptile of the canary grass bending toward the unilateral source of light. When they have taken the coleoptile of the canary grass and they have shown the monochromatic light from one side. So they saw this coleoptile is bending towards the unilateral source of light. And it was concluded after a series of experiments that some substances are produced at the tip of the coleoptile that was responsible for the bending of the coleoptile. And finally, this substance was extracted from this coleoptile and called as auxin, right? So what are the physiological functions? They control the plant cell growth. Okay, so this auxin control the plant cell growth. They cause the phenomena of apical dominance. Okay, so they shows the dominance at the apex. So the phenomena of apical dominance is shown by this auxin hormone. They control the division in the vascular cambium and xylem differentiation. You know that xylem is having the 4 different types of cells. So the xylem differentiation is controlled by this auxin, and they also shows the division in the vascular cambium. They introduce parthenocarpy and prevent the abscission of leaves and fruits. So parthenocarpy, seedless fruits, are developed by the action of this particular hormone, and they also shows to prevent the formation of abscission layer in the leaves and fruits. Horticulture applications, they are used as the rooting hormone in the stem cutting. 2,4-D is used as a weedicide to kill the broadleaf dicotyledonous weeds. So they are used as a weedicide. We can kill the weeds and we can protect our plant, right? So they introduce parthenocarpy in tomatoes. When these hormones are sprayed, auxin is sprayed. So they on the tomatoes, they produces seedless tomatoes, that is called as parthenocarpy. They promote flowering in the pineapple and lychee. Okay, Next is the gibberellic acid. Bacne or the foolish rice seedling disease was first observed by the Japanese farmers. And in this disease, rice seedlings appear to grow taller than the natural plant. So they have extracted this extract from this disease of the plant, that is the foolish rice seedling. And later, after the several experiment, it was found that the condition was caused by the infection from a certain fungus, Gibberella fusiculae. When they have spread, they show that the plant is increasing in their height. So the active substance that was isolated from that fungus was the gibberellic acid, right? Now later the gibberellic acid was extracted from the plant itself. What are the physiological functions of this? It causes elongation of the internodes. Okay, the internodal region of the stem increases in size. It promotes bolting in roseate plants. So some of the plants which is it is having the lesser size of the internode, so it forms the roseate like structure. But when it is spread with the gibberellin, the internodal region increases and they form the bolting phenomena. It helps in inducing the seed germination by breaking the seed dormancy and initiating the synthesis of hydrolysis enzyme for digesting the reserve food material. Okay, so it helps in inducing the seed germination also. Horticulture applications. It helps in increasing the sugar content in the sugar cane by increasing the length of the internode. So if the internode increases in the case of sugar cane plant, then what happens? Okay, the sugar content of the sugar cane increases. It increases the length of the grape stalk also. It improves the shape of apple. Okay, shape of apple, and it is commercially important also for the selling of the apple. It delays senescence, okay, and it hastens the maturity and induces the seed production in the juvenile conifers. Next hormone is the cytokinins. Its discovery through the experimental observation, F. Skoog and his co-worker found that the tobacco callus differentiate when extracted of the vascular tissue. Yeast extract, coconut milk or the DNA were added to the culture medium. So if you add these things to the culture medium, you can see the callus is forming. So this led to the discovery of cytokinin, because these things promote the active division of the cells. Now, what are the physiological functions? They promote the growth of the lateral branches by inhibiting the apical dominance. They help in the production of new leaves, chloroplast, and adventitious shoots also. They help in delaying senescence by promoting nutrient metabolism. And what are the horticulture application? They are used for preventing apical dominance. Okay. The apical dominance was shown by the auxin, and this particular hormone is used to prevent the apical dominance. So it used for the increasing the branching. They are used to delaying the senescence in the leaves also. Next hormone is the ethylene. So it was discovered that the unripe banana, okay, ripened faster when stored with the ripe banana. Some of the ripe banana is kept with the unripe banana. So you see that the unripe banana get ripened very fast. So later the substance promoting the ripening was found to be ethylene. Okay, so some of the substances that coming out from the ripe banana, and it is in the form of the gaseous form, that is the ethylene, right? And that was later discovered that these ethylene gas was coming out from this ripened banana. So what are the physiological functions? It helps in breaking the seed and bud dormancy. Okay, it promotes rapid internode elongation and the deep water rice plants. It also promotes the root growth and the formation of root hairs, and it promotes the senescence and the abscission of the leaves and flowers. So it hastens the respiration rate in the fruits and enhances the fruit ripening as well. Horticulture application, it is used to initiate the flowering and synchronizing the fruit set in the pineapples. Okay, so it is also used in the flowering and synchronizing the setting of the fruits in the pineapples. It induces flowering in mango. Ethaphon is used to ripen the fruits in tomatoes and apples and accelerate the abscission of the flower and leaves in the cotton, cherry, and also in the walnut. It promotes the number of female flowers in cucumbers. And the last one is the abscisic acid. How it got discovered? During the mid 1960s, inhibitor B abscission 2, a dormine were discovered by the 3 independent researchers, and they were later on found to be the chemically similar, where they're after called as ABA, that is the abscisic acid. What are its physiological functions? It acts as an inhibitor to the plant metabolism. It is a inhibitor of the plant metabolism. It stimulates the stomatal closure during the water stress. Okay, when there is a water stress, it induces the closing of the stomata. It also induces the seed dormancy. It keeps the seed in the dormant condition, and when the favorable condition returns, they undergo for the process of germination. It induces the abscission of leaves, fruits, and flowers also. Horticulture application, it induces the seed dormancy in stored seed. Okay. So next question here. What do you understand by the photoperiodism and vernalization? Describe their significance. So photoperiodism refers to the response of plants with respect to the duration of light. And on the basis of the response to the duration of light, plant is classified. How much the time of light is exposed to them. It is classified as a short day plant or a long day plant. Day neutral plant. So short day plants flower when they are exposed to light for a period of less than the critical daylight. Okay. For example, chrysanthemum. It is a short day plant example. Long day plants flower when they are exposed to the light for the period of more than the critical daylight. That is the critical daylight, that for example, radish. And when no marked correlation is observed between the duration of exposure to the light and the flowering response, then the plants are termed as a day neutral plant. For example, tomato. Now, it is hypothesized that the hormonal substance responsible for the flowering is formed in the leaves, subsequently migrating to the shoot apices and modify them into the flowering apices. So this leaf apices is generally modified to the flowering apices, and this occurred due to some of the hormones. So photoperiodism help in studying the response of the flowering in the various crop plants with respect to the duration of the exposure to the light. Next is the vernalization. It is the cold induced flowering in the plants. When you gives the cold treatment and it induces the flowering in the plant, then it is called as vernalization. So in some plants such as winter varieties of wheat and rye and the biennials such as carrot and cabbage, exposure to the low temperature is necessary for flowering to be induced. Okay, so in these plants, it is required to the exposure of the plants to the low temperature. So the winter varieties of rye and wheat are planted in autumn. So they remain in the seedling stage during the winter and flower during the summer. However, when these varieties are sown in the spring, they fail to flower. So similar responses is seen in the cabbage and the radish also. Why is abscisic acid is also known as a stress hormone? It is also called as a stress hormone. So abscisic acid is called stress hormone as it induces the various responses in plant against the stress conditions. It increases the tolerance of the plants toward the various stress. Okay, so it helps the plants to tolerate the stress. It induces the closure of the stomata during the water stress. When there is water stress, it causes the closing of the stomata, and it promotes the seed dormancy and ensures the seed germination during the favorable conditions. It also helps in seed withstands in the desiccation, and it also helps in inducing dormancy in plants at the end of the growing season and promotes abscission of leaves, fruits, and flowers. So that is why it is called as a stress hormone. Next question, both growth and differentiation in higher plants are open. You have to comment on this. So growth and development in the higher plant is referred to as being open. This is because various meristems having the capacity to continuously divide and producing the new cells, and the present at different locations in these plant body. So meristems are present at the different location and they divide and redivide. So that is why both growth and differentiation in higher plants are open. It is occurring at the various points of the plants. Both short day and long day plants can can flower simultaneously. Give in a given place. Explain. So flowering plants, flowering responses in the short day plants and the long day plant is dependent on the duration for which these plants are exposed to light. And in the short day plant, the long day plant can flower at the same time, provided they have been given an adequate photoperiod. And the next question here. Which one of the following growth regulator would you see if you are asked to induce rooting in a twig? So induced rooting in a twig is done by the auxin. Okay. And quickly ripening of a fruit, this is done by ethylene. Delay in the leaf senescence is done by the cytokinin hormone. Induced growth in auxiliary buds is done by the cytokinin. And bolt or rossity plant, that is the gibberellic acid, induce immediate stomatal closure in the leaves is done by the abscisic acid. Would a defoliated leaves plant respond to the photoperiodic cycle? A defoliated plant will not respond to the photoperiodic cycle. Why? It is hypothesized that the hormonal substance responsible for the flowering is formed in the leaves and subsequently migrating to the shoot apices and modify them into the flowering apices. Therefore, in the absence of leaves, light perception would not occur and the plant would not respond to the light. Okay. So here comes the last question. What would be expected to happen if GA3 is applied to the rice seedling? So if GA3 is applied to the rice seedling, then the rice seedling will exhibit internode elongation and increase in the height. And next question was dividing cell stop differentiating. If a dividing cell stop differentiating, then what will happen? Then the plant organs such as leaves and stem will not be formed. And the mass of undifferentiated cell, that is called as a callus, will be formed. Next, what is the question? A rotten fruit get mixed with the unripe fruit. If a rotten fruit get mixed with the unripe fruit, then the ethylene will produce from the rotten fruit. Okay, and it will make the unripe fruit to the ripened one. Last one, you forget to add cytokinin to the culture medium. If you forget to add cytokinin to the culture medium, then the cell division, growth and differentiation will not be observed. Okay, because cytokinin is very, very important for the division of the cell. So with this, we end our session here. We will meet with a new session. Till then, thank you.